Alright, so I'm gonna start here. Alright, so yesterday we were talking about meiosis, and I want to just go back to this to give you the big idea here. This picture. So the DNA replicates here, and so you have replicated chromosomes, and that's an interphase. And then remember, there's the first division of meiosis where you go to 1 to 2 cells, and that's where the pairs of chromosomes separate. And then the second part of meiosis, you go from 2 to 4 cells, and the sister chromatids separate. And so you end up having half the DNA as your original cell. So what we talked about yesterday was literally the movement of the chromosomes and what happens to the chromosomes to go from 1 to 2 cells so that your homologous chromosomes are in opposite cells. So what happens here is when they line up, remember the spindle attaches and pulls the homologous pairs to opposite sides of the cell, and then the cell splits and you end up with 2 cells. That's what this picture was showing you here, is that remember prophase, they come together with their partners, and that's called a tetrad. They cross over, the point at crossing over is called chiasmata, and they switch DNA at that crossover point, remember from the point of crossing over to the end of the arm. They line up with their partners down the center of the cell during metaphase. The partners, the homologous chromosomes, begin separating during anaphase due to there's only one spindle attached to each chromosome, and so this spindle attaches here and pulls it to this side of the cell. The homologous chromosome over here, it only has one spindle over here, and it pulls it to the opposite side of the cell. And so then we looked at telophase, where now you have one of each kind of replicated chromosome on either end of the cell, and you have a nuclear envelope reforming, and then cytokinesis starting where that indentation is occurring. But this picture is still telophase. Is this cell haploid or diploid?
Diploid.
This one is diploid. So this is still, even though you have a haploid set here and a haploid set here, it's still one big cell, so therefore makes it diploid. Once that cytokinesis happens and it splits into 2 cells, these 2 cells are now haploid, right? These cells are now haploid, containing one set of chromosomes, one large, one medium, and one smaller, okay? And so that's what we have here. So this is what we're on. So we're on meiosis 2. Notice that right after cytokinesis for meiosis 1, it splits into 2 cells, it immediately enters into prophase 2. So prophase 2, let me point this out, that notice here in telophase that the nuclear envelope reforms. Then once it splits in 2 and then enters prophase, the nuclear envelope disintegrates again. So it reforms, only yet to break it down right away. And so it breaks down, and then what happens in prophase here is what happens in prophase in all the other prophases that we've talked about. So the spindle is forming, the nuclear envelope is breaking down. And so there's your prophase. Then, in this case here, in metaphase, they all line up single-filely. This is different than metaphase 1. Metaphase 1, they lined up with their partners so that the partners could be separated to opposite cells. In this cell, there are no partners. There's one of every kind of chromosome, so they all line up. The purpose of this second cell division is to separate the sister chromatids. So they all line up. And so this side is made out of one piece of DNA, and this side is made out of another piece of DNA, and they're eventually going to separate to opposite sides of the cell. So what we see here is the chromatids separating through anaphase, and in telophase then you have one of each type of chromosome on either side of the cell. This still is diploid until the cell membrane begins to indent some more, and it actually divides. So this actually doesn't show you the end cells, but this cell here, once it divides, you're going to have 2 cells eventually at the end, and those two cells will have in the nucleus the 3 pieces of DNA, and so therefore they only have one of every kind of chromosome, and then these cells will divide into 2, and you end up with 2 cells in the nucleus having 3 pieces of DNA, exactly half of what you started out with. So you end up with the 1, the 2, the 3, and the 4 cells at the end. All right, and so that is meiosis 2. So, so I just verbally explained this. So the rest of the notes here as we go along just talks about each of the phases that I just went over. So prophase 2, and so meiosis 2 is very similar to mitosis. In prophase 2, the spindle apparatus forms. The chromosomes start moving towards the metaphase plate. Here. At metaphase 2, the sister chromatids are arranged at the metaphase plate, and because of crossing over, the 2 sister chromatids of each chromosome are no longer genetically identical, and you have kinetochores, these microtubules, attached on either side of the cell, so extending from opposite, each side of the chromosome, I should say. So what we're looking at here, they're lined up single-filely. They're showing you here that because crossing over occurs, they've already exchanged DNA back in meiosis 1, and so this chromatid on the left side is not genetically the same as the chromatid on the right side, and so therefore, that's what it means by not being genetically identical. And then they have a microtubule attached. Each chromosome has a microtubule attached from either side of the cell. That's important so that that separates the chromatids to opposite sides of the cell in anaphase. So in anaphase, the sister chromatids separate. The sister chromatids of each chromosome now move as 2 newly individual chromosomes towards opposite poles. And so that's what we see here. And then eventually we get to telophase 2 and cytokinesis. In telophase 2, the chromosomes arrive at opposite poles. The nuclei form again, and the chromosomes begin decondensing. So they become unraveled again, forming chromatin. Cytokinesis then separates the cytoplasm, and so you end up with 2 cells, or 4 cells total. So at the end of meiosis, there are 4 daughter cells, each with a haploid set of unreplicated chromosomes. And they're genetically, each daughter cell is genetically distinct from all the other ones, as well as the parent cell because of crossing over, because they exchange genetic information. And so that's what we see here. And then, again, they divide, and then you have your 4 cells at the end. So what I'd like you to do is, I want you to look at the 2 pictures. They're kind of divided into, so there's meiosis 1, and this picture is meiosis 2. So you have those 2 pictures in your notes. Because what I'm going to have you do is look at each, starting with meiosis 1, and underneath each picture, I'd like you to do a couple of things. The first thing I'd like you to do is to look at the picture and tell whether or not the cell is haploid or diploid at that particular stage. So haploid would be just N, diploid is 2N. So tell whether it's haploid or diploid. And the second is I'd like you to write the number of chromosomes in the cell. And be specific, are they replicated chromosomes or single? All right, so those are the 2 things. And when I say number of chromosomes, I wanted you to literally the picture, the literal picture, so not like what human cells would have, but that particular cell. So underneath, that's what I'd like to do each picture. So start with the picture, starting out with meiosis 1. So you have to flip back on page or 2. And see if you we'll go over it.
No.
Actually, are you going to go out of coffee? It's making me so diploid is 4 generations.
So in meiosis, thanks for that meiosis. So aren't you solid? Or would this be different? Because they never divided. And then oh, there's only three. Like, what are she split?
Single chromatids is your area.
I think that's the best thing ever you say. Like, he makes jokes often, and sometimes I always laugh. But sometimes they're like a mediocre, classic dad jokes. I'm like, that was a killer one. I figured it. Okay. Each six single or three pairs. What about your brothers and sisters?
What?
With the Mary question at the end.
Or is that on the
No.
Okay, once you get past here,
They're all happy.
like,
She said something when they were talking. She was like, Anaphase or something. Because they're not getting what I got. So I don't know if that was a mistake, but
nope, you know,
like, no one would have it. And the bigger they go,
some are replicated after this
yes.
here, replicated. You know what they are?
It needs to be gone. Yes, because they're so weak. Oh no, I'm famous. Oh no, people are actually watching that one. Okay. So all of that. What is haploid? What is this? Question. Single. So this is the parental three singles. And what happens when you get down here? Sister. Or homologous. So when we get so this is haploid.
Homologous would mean tetrad.
Three pairs. It's not three pairs.
These are
Three replicated chromosomes.
these are
One
homologous.
chromosome. So this thing is a copy of the seed. Oh, sorry. So it's just one chromosome made up of two. Right? So this is three replicated.
Too bad they're not single.
So the thing, so when you go from here, it's three rower area. But there's still three replicated chromosomes. So there's something different. These are these pages here, but the tissue comes when now the chromosomes separate. So this is a, so yeah, so when they separate, now this is one replicated chromosome, this would be two separate. So it's just single, that's because yes, because this isn't and so that will go back to that would still be diploid, yeah, until it's split. Okay. So what haploid still creating? Yeah. And then this one's diploid because now there's 6 single instead of 3 replicating. And it's
That's not true.
so it's not
That's not a person.
pairs is
I guess I have to.
yeah.
All right, let's okay, so for prophase, prophase 1 here, this cell is a diploid or haploid?
It's diploid.
This cell is diploid. How do we know when looking at this that it's diploid versus haploid? Nathan?
You got the two said each chromosome has a pair.
That's right. Each chromosome has a pair, a match. All right. So we have pairs here. So the long, these two go together, these two go together, and these two go together. So how many chromosomes are in this cell? These are six chromosomes. So this is a diploid cell you should have. So we represent that by 2n. If you were to write the equation, guys, if you were to write the equation, this is a diploid cell, you'd say 2n equals what? 6. All right, 2n equals 6. So we have a total of 6 chromosomes, and the 2n means that it's 2 of every kind of chromosome. That's the diploid part, the pairs. So then what would n be for this cell? 3, because there's 3 types. All right, and then we can distinguish them by length. All right.
One question. Like those chromosomes that are color coded blue and red, does it mean like one from father and one from mother?
Yeah, that's why we have pairs. Is one from mom, one from dad, and they're color coding them. The matches different colors to make it easy to tell when crossing over occurs. So you can see that now the blue chromosome has part of the red chromosome's DNA. So that's kind of the purpose of illustrating them. But absolutely, you have one from mom, one from dad. Okay. Okay. If they color coded these guys the same, all the same color, when crossing over occurred, like in this picture here, you wouldn't be able to necessarily tell, oh, this has a new piece of DNA on it. So having the red part of the arm just helps you to do that. All right, so this is diploid. You have 6 replicated chromosomes, right? They're replicated. And so then here, from here to metaphase 1, the chromosomes are now lined up, but has the chromosome number changed or the status of the cell at all? No. So this cell, in terms of diploid and haploid and the number of chromosomes, is the same as this cell. So this is still diploid, having 6 replicated chromosomes, right? And then here, the chromosomes are moving to opposite sides of the cell, the homologous chromosomes. But again, all those same chromosomes are still in this one cell, so the status of the cell hasn't changed. So you should have also, for this cell, it should be diploid with 6 replicated chromosomes, right? Okay, so then we move to, let me go to mine's on different, this picture. So telophase and cytokinesis. So this is still one cell, so therefore we should have that this is diploid, and there's still 6 replicated chromosomes in there. It's not till the very end where it just pinches off and the cytoplasm is separated that we end up with 2 cells that now begin prophase 2. These 2 cells, do they have pairs in them anymore of chromosomes? No. So these cells are considered haploid. These are haploid, and they have 3 replicated chromosomes. All right, so 3 replicated chromosomes, 1, 2, 3, and 1, 2, 3 here. So then here, the chromosomes start lining up down the middle, so they've arranged themselves differently, but there's still 1, 2, 3 replicated chromosomes, and it's still a haploid cell for both of those. All right, so then it becomes tricky in anaphase 2. Anaphase 2, so you have these chromosomes here that are replicated, and remember the replicated chromosomes are made out of two sister chromatids. So if my arms are the sister chromatids, so this copy is an exact copy of this copy, and they're connected at the centromere. And so this is your replicated chromosome. This is one replicated chromosome here, one here, one here. What happens here when the chromatids separate? Are they part of the same chromosome anymore? No, they are not. And they are considered their own separate little chromosome now. And so therefore, this cell has how many chromosomes? 6 single chromosomes. Now, would I consider this a haploid or a diploid cell at that point?
Diploid.
It is. It's diploid. It has 2. So, like, this, let's say this is chromosome 1, 2, and 3. It has now 2 single chromosome number 1s and 2 single chromosome number 2s, and so on. And so this is diploid, which then here, when we go to telophase, when it's still in telophase, we have our single chromosomes. They're starting to unravel and becoming chromatin again, but they're on opposite sides of the cell, the haploid sets, but this is still one big cell. So even though these guys are on one side of the cell, these guys are on another side of the cell, it's still one cell, so therefore you would still have your 6 single chromosomes in that cell, and it would still be diploid. It doesn't become a haploid until cytokinesis finishes, when this actually splits. These cells that I drew here, these cells, all of these guys, would all be haploid, and they would all have 3 single chromosomes, and actually, realistically, the chromosomes would unravel and become chromatin. Hi.
Is it okay if I take pictures for your book?
Sure, sure. All right, does that make sense? How'd you guys do on that?
I have one question.
Yeah.
Like, if you were to look at an actual cell, it has, so every, like, even somatic cells, they all have 23 pairs of chromosomes, right?
23 pairs, yeah.
So when they divide, they also have to divide the 23rd pair, even though they don't need time.
Say that again?
Wait, so somatic cells, which are not, don't take any part in meiosis.
Right.
So when they divide, they also divide the 23rd pair, which are sex chromosomes. And gametes are the only cells that use the 23rd.
No. Gamete, every cell uses the 23rd pair has genes on it just like any other, like, so you have an X and a Y in your somatic cells. Though that there's genes on your X chromosome and there's genes on your Y chromosome that make proteins for you, and the proteins that they make give you maleness. So there are genes on the Y chromosome that only males have that make proteins that only males can make because they're the only ones that have that gene. So it's really the presence of that Y chromosome that determines maleness. And if you don't have a Y chromosome, by default you're a female.
Oh, that makes sense.
All right, does that make sense?
Yeah.
All right, and so it's in every cell of your body. It's just egg and sperm cells is the vehicle to give those chromosomes so they can match up with the egg and the sperm to give them to the kids.
So after the egg is fertilized and it has code 46, then it just goes through regular mitosis.
That's correct. All right, that's exactly right. So that's why, because we started out with only one cell, we're all one cell big. So this is why mitosis just copies all your DNA and divides, and so each new cell has those same 46 chromosomes. So this is why, like, you could go in, all of the DNA that makes you is in any cell in your body. So you can take a cell from your eyeball or a cell from your skin, and there's the same DNA in it. Okay, not all the DNA is coding for proteins in all the same cells, but it's all there. Absolutely. Any other questions on this? No? Alrighty. So that is mitosis. So the next little bit here does a comparison of mitosis with meiosis. So we've already talked about mitosis and we've talked about meiosis. And so it says, Meiosis blank the number of chromosome sets. So it conserves the number of chromosome sets, producing cells that are genetically identical to the parent, which is what Roman was just talking about. And meiosis, on the flip side, reduces the number of chromosome sets from 2 to 1 set. So we have 2 in every of our somatic cells, goes to 1. It says that differ genetically from each other and from the parent cell. How does, at the end of meiosis, when you get those 4 cells, how is it that they differ genetically from one another? What contributes to that? Crossing over, absolutely. And so then you separate the sister chromatids in meiosis 2. And so meiosis 2 and mitosis are basically the same type of problem, or process, I should say. So there are 3 things that stand out as being different in the process of meiosis than in mitosis. And these 3 things are the synapsis and crossing over in prophase 1. So remember, the homologous chromosomes find one another in prophase 1 and physically connect. They cross over and exchange that genetic information. The way that they line up at the metaphase plate, there are paired homologous chromosomes instead of individual replicated chromosomes. And then lastly, in anaphase and meiosis 1, the homologous chromosomes separate instead of sister chromatids, and so those guys separate. And so that, and the purpose of that is because, remember, in meiosis we want to reduce the chromosome set. So they line up in pairs so that each pair, one of each one, goes into a separate cell, and you don't get pairs of chromosomes interacting with sperm cells. So that when the sperm fertilizes an egg, then you get pairs. And so this picture here shows you this process. And so it just I like this because it just gives you a basic overview of both mitosis and meiosis to show the difference there. And so notice here, this is where we're starting here, the parent cell at the beginning. The parent cell at the beginning looks the same and has the same amount of DNA at the beginning for mitosis, the cell at the beginning of mitosis, or meiosis. So in this organism here, here's your single pieces of DNA. So they have 3 pairs, so this is a diploid cell, and in interphase, remember, the DNA replicates, so now we have 6 replicated chromosomes. They condense and form chromosomes, the DNA. And then in mitosis, all the chromosomes line up single-filely, so that the sister chromatids separate, and you end up with 2 cells that have 2 of every kind of chromosome, 2 diploid cells that are exactly like the first parent cell. Now, we don't want that in our 4 cells at the end of meiosis. So this same cell, where were these cells that are going to go through meiosis and go in this direction? Where do we find those cells? Where would I find this cell that's going to go through meiosis? They're going to make gametes. This cell is going to divide through meiosis to make gametes. Where do I find that cell that's going to divide there, Sage? The gonads, which are ovaries in females and testes in males. All right, so that's exactly right. So that's where we find these, and they are the only cells in our body that will divide by meiosis. And so the same thing happens where the DNA replicates here, but what's different that we just wrote about is that they find their partners. All the homologous chromosomes find their partners, and they do the crossing over thing. So that's unique to meiosis. Then, when they first line up, mitosis, they lined up single-filely. Here they're with their partners. That's because in meiosis 1, we can separate the pairs into 2 opposite cells. So no longer do we have pairs of chromosomes in these 2 cells. So that's the second difference, is the way that they line up. And so then, then these guys, I'm not showing you here, these guys then will line up. I wish they showed you it lined up single-filely, because that's why I said meiosis. It tells you meiosis 2, where you go from 2 cells to 4 cells. It's like mitosis, because they all line up, all the replicated chromosomes line up single-filely. chromatids separate and you end up with 4 cells at the end. And you can see here that all of these 4 cells are different with the color coding here. Obviously yours is not color coded, but you can see that none of them have the exact same combination or the way that crossing over occurs. So neither one of these 4 cells are identical to one another. So that crossing over provides genetic diversity. And so those are your daughter cells that are all haploid. Notice one of each kind of chromosome, exactly half of what you started out with. Okay? And so that's our comparison there. And so you have a chart, and unfortunately the headings of that chart I didn't notice was on the bottom of the page, and then the rest of the chart is on the other side. So I would, if I were you, rewrite the headings so you can just look at it that way. Just a nice, neat chart to compare and contrast mitosis and meiosis. So all of these we've already talked about. I'm not going to read through there with you, but I do want to just point out, because sometimes this people get confused with the number of divisions. So mitosis is 1 division, meiosis is 2. So what that means, mitosis, like in humans, we start out with a cell with 46 chromosomes, we replicate those, and we end up with 2 cells with 46 chromosomes. This is considered 1 division, even though, because people are always going to go, well, isn't there's literally 2, I guess, right here, but they consider from 1 to 2 cells 1 division, okay? People want to count these things here, okay? So that's 1 division of mitosis. In meiosis, you have meiosis 1, where we go from 1 to 2 cells, and then you have meiosis 2, which is 2 to 4 cells. These are considered 1 division, the 2nd division, okay? And so that's where that comes into play, okay? And that is mitosis and meiosis. You guys, I think I'll stop here. All right, thank you. You guys hung with me. All right, so we just had a little bit, probably 15 minutes or so of that, but I'll stop here for the day.